Mitogen-activated protein kinase, shortened as MAPK, is a type of protein kinase that is specific to the amino acids serine and threonine. MAPK was originally called extracellular signal-regulated kinases (ERKs) and microtubule-associated protein kinase (MAPK). After many additional targets for phosphorylation by MAPK were later found, the protein was renamed mitogen-activated protein kinase (MAPK). The MAPK cascade will result in gene target transcription in the nucleus. RAS activates a MAP-kinase signaling module. An activated RAS protein will result in activated MEKK, MEK, and MAPK. This MAPK will cause change in protein activity and change in gene expression in the nucleus. What are P90RSK and CFOS? Ribosomal acid kinase, also known as RSK, is a family of protein kinases involved in signal transduction. There are two subfamilies of RSK, which are P90RSK and P70RSK. CFOS is a proto-oncogene. A proto-oncogene is a normal gene which, when altered by mutation, becomes an oncogene that can contribute to cancer. It has an important role in signal transduction, cell proliferation, and differentiation. G-protein-coupled receptors, also known as GPCRs, are integral membrane proteins that have a common 7 transmembrane segment structure. These are the example of GPCRs. G-proteins, also known as guanine nucleotide binding proteins, is a family of proteins that act as molecular switches inside cells and are involved in transmitting signals from stimuli outside the cell to its interior. G-proteins consist of alpha, beta, and gamma units. This is a diagram of G-protein activation-inactivation cycle. The activation-inactivation cycle of G-proteins. In the resting state, alpha is bound to GDP. Ligand binding induces a conformational change in the receptor such that the cytosolic domain of the receptor interacts with the G-protein and stimulates the release of bound GDP from alpha subunit, and GTP binds to the alpha subunit. The activated GTP bound alpha subunit then dissociates from beta and gamma, which remains together as beta-gamma complex. Alpha subunit moves along the inner membrane and interact with another membrane-bound protein, primary effector, adenyl cyclase, to elicit an intracellular response. The activity of the alpha subunit is terminated by hydrolysis of the bound GTP. The now inactive alpha subunit then reassociates with beta-gamma complex, ready for the cycle to start anew. This diagram shows the full process of activation-inactivation cycle of G-proteins. These are the examples of G-proteins and their effector. This diagram shows the signal amplification by second messengers in the intracellular effects. And these are the structure of ATP, CAMP, and AMP. This is the cyclic AMP second messenger system. This diagram shows that GPCRs regulate adenyl cyclase by using inhibitor in order to control the amount of CAMP produced. This shows another example of GPCRs that regulate ion channels, which is muscarinic acetylcholine receptor that results in the slowing of heart rate. This diagram shows mechanisms by which an activated receptor stimulates the enzymatically mediated breakdown of PIP2 to yield IP, while this diagram shows the inositol triphosphate IP3 signaling pathway. That is all from us. Thank you.